Hi everyone, in this video module I am going to talk about Plasmodium. If you recall the classification of protozoa, we had kept Plasmodium in the category of non-motile protozoa. Okay, it's a non-motile protozoan. It's the causative agent of malaria. Alright, now the important species name you need to remember, for instance Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae and Plasmodium ovale. Remember guys, vivax is the most common cause in India. Alright, then remember F for falciparum, F for fatal. Falciparum causes fatal disease. It, in other words, causes the most severe malaria. It causes the most severe malaria. Alright. Now, ovale is not found in India. It is not found in India. Remember that. Ovale begins with O. Remember, O reminds of 0. 0 means absent, not found in India. Okay, just remember like that. And there's one more species. Just know the name, Plasmodium knowlesi. This is actually a parasite of monkeys. But occasional cases of human malaria because of this have been reported from Asia. We still know less about it. It is called as knowlesi. That's not the reason, by the way, the species has been named so. Just remember knowlesi, we know less about it. It's still being studied. But these four are the most important species which infect humans. Vivax, falciparum, malariae and ovale. Now, there are certain, fancy terms to describe the severity of the disease and when the fever spike occurs, which you need to know. For instance, in case of Plasmodium vivax, the kind of malaria which is described is known as benign tertian malaria. Benign means what? The disease is less severe, not a very severe disease it is. And tertian means fever spike occurs on day 3, that is after 48 hours. Okay, that is after 48 hours. Then we have Plasmodium falciparum which causes malignant tertian malaria. Malignant means what? It means severe disease. Tertian again means, fever spike occurring after 48 hours, that is on day 3. I already mentioned before, F for falciparum, F for fatal, it causes the most severe kind of malaria. Then Plasmodium malariae, it causes quartan malaria. M and Q underline, remember M, C and Q. Alright, now M of MCQ, M for malariae, C for nothing, Q of MCQ, Q for quartan. Then finally we have got Plasmodium ovale which causes ovale tertian malaria. It causes ovale tertian malaria. Next, what we need to know about this parasite is, we are going to talk about its life cycle guys, for which you need to understand certain terms. For instance, the definitive host. I am going to use the short form DH for definitive host. Alright, the definitive host is female Anopheles mosquito. Alright. Now, definitive host of a parasite is the one, it is a host in which sexual cycle takes place. Sexual cycle of the parasite takes place, and this sexual cycle which takes place in female Anopheles mosquito is known as sporogony. Now, this word sporogony implies that during sporogony, sporozoids are formed. These sporozoids are present in the mouth parts. They are present in the mouth parts of the mosquito. Sporozoids which are being formed in the mosquito, guys, these are the infective form. These are the infective form for humans. So when the female Anopheles mosquito, which has got sporozoids in its mouth part, when this mosquito bites humans, it is going to inject sporozoids into the bloodstream of human. So for humans, the infective form is going to be the sporozoids. Now, why the male Anopheles mosquito is not the definitive host? Because it is the female mosquito which needs a blood meal for egg maturation. Okay, not males. Not the male mosquito, because the female mosquito needs blood meal for egg maturation. For egg maturation. Alright, then what is the intermediate host? See, there are very few parasites in parasitology where man serves as the intermediate host, and this is one example, malaria. Intermediate host, I am going to use the short form IH for intermediate host. Intermediate host is man. Intermediate host of a parasite is a host in which asexual cycle takes place. Asexual cycle of the parasite takes place. Now, the asexual cycle which takes place in man, that is the intermediate host, is known as schizogony, during which schizons are formed. The moment somebody says the word schizont, what should you think of? What should come to your mind? What is a schizont? A schizont is nothing but a multi-nucleated structure. How is it formed? How the multiple nuclei, happen to appear? That all we are going to discuss in a while. Now let's see what happens when the infective mosquito bites a human. Now the mosquito, of course the female Anopheles I am referring to, the mosquito when bites humans, what happens? Sporozoids are injected into the bloodstream. Okay, sporozoids are injected into the bloodstream. Now these sporozoids are like this, spindle shaped. Okay, this is the nucleus. Spindle shaped sporozoids. Alright, now they are present in the bloodstream, they can go anywhere, but their most favorite site of infection is what? It is the liver. So these sporozoids reach the liver, and in the liver what will happen? In the liver a kind of cycle will take place known as pre or exoerythrocytic pre or exoerythrocytic schizogony. It is called as pre or exoerythrocytic schizogony because this cycle takes place or it occurs before the RBCs are invaded. Before the RBCs are invaded. And of course, it is, we are talking about what is happening in man. Remember, in man, schizogony occurs. So, this cycle is going to take place in the liver. It means some schizons are going to be produced in the liver. Okay. So, what happens in the hepatocytes? In the hepatocytes, now imagine this is a hepatocyte. An over schematic diagram this is, guys. This is a hepatocyte. What happens? See, suppose this light green colored thing is the sporozoid. Okay, this sporozoid is going to round up, right, like this to form what is known as early trophozoid or the ring stage. Early trophozoid or the ring stage. Then this will undergo few more changes. It will mature into what is known as late trophozoid. Okay, then late trophozoid, that is LT. Early trophozoid is ET, late one is LT. Alright, then what will happen? Then nuclear division will take place. Alright, multiple nuclei therefore will be produced like this, and this multi-nucleated structure, guys, is known as what? It is known as, this multi-nucleated structure is known as hepatic schizont, because it is being formed in the liver cell, right? Subsequently, what will happen? Around each of this nucleus present in this schizont, around each nucleus, membrane will be laid, which I am designating in orange color like this, like these orange circles. Alright, and all these now orange circles containing the nuclei, all these are known as merozoites. The moment merozoites are formed, what will happen? These merozoites, they will be released out. They will be released out by the rupture of the hepatocyte. Okay, rupture of the hepatocyte, and they merozoites are now going to circulate in the bloodstream. The merozoites circulate in the bloodstream. Now, some of these merozoites which are floating or circulating in the blood, some merozoites are now going to invade the RBCs and initiate or start the erythrocytic cycle, also known as the erythrocytic schizogony. So, what is going to happen over here? In erythrocytes, schizons will be formed. Erythrocytic schizogony. Erythrocytic schizogony. Now, imagine, so what is going to happen? If suppose this is an RBC, okay, this is an RBC, I had used the orange color for merozoites, and the green dot represents the nucleus, okay. So, I will use the same color coding. Imagine this is the merozoid which has entered inside the RBC. Now guys, from this point onwards, the stages, the morphological transformation, from merozoid to different forms is going to be the same as we saw in the liver cell. Remember, in the liver what was happening? It was the sporozoid which invaded the liver, and subsequently early trophozoite followed by late trophozoite followed by merozoites, followed by schizon, followed by merozoites. Okay, now the same sequence we are going to follow. The only difference is that here the starting point is going to be the merozoid. So, what is going to happen? This merozoid is going to change into what is known as the early trophozoite or the ring form. Okay, now this is nothing but this one. This is referred to as the early trophozoite or the ring form or the ring stage. As you can see, I have drawn, this is the nucleus. This nucleus is displaced to the periphery because there is a huge cytoplasmic vacuole. Okay. Now, let me represent the vacuole by this silver color. So, this is a huge vacuole. Right, this nucleus is pushed to the periphery, and what picture it is giving off? It is looking like diamond on a ring. So this is the classical description for this ring stage, diamond on a ring. Diamond on a ring. Now next, it is going to undergo a certain more maturational changes, and then we are going to call it what? Then we are going to call it the late trophozoite. So this is going to be the late trophozoite. Now subsequently, Recall what did we discuss in the liver cell? Sporozoid to early trophozoid to late trophozoid to schizon to merozoid. So here also after late trophozoid, guys, the nuclear division is going to take place and multiple nuclei you can expect. Now this entity, this structure is referred to as what? It is referred to as the erythrocytic schizon. The erythrocytic schizon. And next what is going to happen? Next again a nuclear membrane is going to be a membrane, a cell membrane is going to be laid down around all these nuclei. Let me represent the membrane in light blue color like this, and all these light blue structures with the green nuclei, they are referred to as merozoids. Merozoids. Guys, so far the patient is not going to have any symptoms. Okay, no fever, nothing. Right? The symptoms, that is fever, chills, rigor and all, will start from this point onwards the moment merozoids are produced in RBCs, because now these merozoids are released out by the rupture of RBCs. By the rupture of RBCs. Okay, and this will herald the onset of the clinical symptoms in the patient. This heralds or starts the onset of clinical features in the patient. Now guys, these merozoids which are released by the rupture of the RBCs, these merozoids invade other RBCs and restart the erythrocytic cycle that is erythrocytic schizogony. Alright, and the same sequence of events will follow, and some of the merozoids, guys, some merozoids transform into male gametocyte and female gametocyte. Okay, female gametocyte is known as macrogametocyte and male gametocyte is known as microgametocyte. Now guys, this is very important. These gametocytes are the infective stage. They are the infective stage or the infective forms for the mosquito. At this point of time, if a female anopheles bite this particular infected individual in whose blood these forms are present, micro and macro gametocyte, this mosquito will pick them up, and then these gametocytes are then going to undergo certain changes in the mosquito, right? If these micro and macro gametocytes, if these are not picked up by the mosquito, then they disintegrate in some time. Alright, in a few days. Now let us see if at all they are going to be picked up by the mosquito, what will be their fate or destiny in the mosquito. So in the mosquito, remember, in the mosquito what will happen? In the mosquito, the sexual cycle takes place known as the sporogony. Sporogony occurs wherein what will happen? The microgametocyte and the macrogametocyte which were picked up by the mosquito during blood meal, respectively they change into microgamete and macrogamete. Now these fuse to form zygote. Okay, then this zygote will transform into a structure called as ookinete. Remember, kineto means what? It means motion. That a kinetic Honda brand is nothing, that the name kinetic Honda is because of its, the motion of the vehicle. So, kineto word means motion. So, this ookinete is a transient morphological form. It's a transient motile. Okay, kineto means motion. So, this is a motile form. It's a transient motile form. Remember, we are saying that Plasmodium is a non-motile parasite. So, do keep in mind that ookinete is just going to play its role for a teeny weeny time only during the life cycle. Okay, it's a transient motile form. Ookinete once forms, it gets converted into oocyst, alright, and this oocyst then multiplies to give rise to multiple spindle shaped sporozoids. Remember, we are discussing sporogony. Okay, sporozoids, these are then released into the body cavity of the mosquito. They are released into the body cavity of the mosquito, and some reach the mouth parts. Some reach the mouth parts, and once these reach the mouth parts, okay, they undergo some more maturation, and then they become competent. This mosquito carrying sporozoids in the mouth parts becomes competent enough to bite another human being and restart another another sort of cycle. So this is about the life cycle of malaria or Plasmodium. Remember, the life cycle of Plasmodium is referred to as what? We call it as cyclopropagative life cycle. Cyclopropagative life cycle. Basically, guys, this descriptive term tells us what? See, there are morphological changes happening. Sporozoid changing into early trophozoid, late trophozoid, schizont, merozoids, and in case of RBC cycle, additionally gametocytes are getting formed. And it is not just morphological transformation, it is also increase in number of the parasite. Remember, schizont is a multinucleated structure, and around each nucleus membrane is being laid down. So now merozoites are and thousands of these nuclei are formed, and after the membrane is laid down around every nucleus, and this way multiple merozoites are released, so the number is also increasing, right? So why am I emphasizing this so much is the fact that there is another parasite called as Wuchereria bancrofti, whose life cycle is called as cyclodevelopmental. That we'll discuss over here, because this is a frequently asked question. Which cycle is cyclopropagative and which parasite cycle is cyclodevelopmental? In case of Wuchereria bancrofti, which causes lymphatic filariasis, there the parasite undergoes only morphological transformation and not multiplication. Alright, but here, apart from morphological transformation, the number is also increasing. Okay, so that is the whole principle. Now guys, the typical symptoms of malaria include fever, chills, extreme headache, rigor and all, and the worst or the severe kind of malaria is caused by the species falciparum. And why is it severe? Because it is associated with a lot of complications, because Plasmodium falciparum does not spare any kind of RBC. It infects all the stages of RBCs, whether mature RBCs, immature RBCs, it doesn't spare any kind of RBC. So next we need to know about the complications of falciparum malaria. The complications of falciparum malaria. Remember, 1, 2, 3. PCB, pernicious malaria. Pernicious malaria means parasitemia more than 5%, meaning out of every 100 RBCs, more than 5 have been invaded by the parasite. Parasitemia more than 5%. C stands for cerebral malaria, which is characterized by encephalopathy. B stands for black water fever. Guys, this is characterized by intravascular hemolysis. Now, black water fever, guys, it doesn't happen on the first attack by Plasmodium falciparum. Okay, it does not. It's basically a hypersensitivity reaction, and it occurs on subsequent attack by Plasmodium falciparum. So it does not occur after first infection. Instead, it's a hypersensitivity reaction. It's a kind of hypersensitivity reaction which occurs, which not always occurs but can occur after the on second attack. On second attack by Plasmodium falciparum. Called as black water fever because it is characterized by the passage of black smoky urine, metabolic acidosis, algid malaria. Questions are frequently asked on this. Algid malaria, which is nothing but malaria plus shock. Which is nothing but malaria plus shock. Then hypoglycemia has got a very poor prognosis. Hypoglycemia. Alright. So this is about the complications of falciparum malaria. Then a couple of words about another complication known as splenic rupture. Remember, splenic rupture is it's a very rare complication of malaria. It sounds, it sounds, obviously it's a very dangerous complication. And when I say dangerous, do not think that the most common cause is going to be again falciparum. No, let the other species also do some job. The most common cause of this rare complication is which species? The MCC most common cause is actually Plasmodium vivax. Okay, Plasmodium vivax. In itself, it's very rare. It's a very rare complication, and if it happens, then vivax is a more common cause than falciparum. In falciparum also it can be seen, but vivax is more common than falciparum. Then also remember nephrotic syndrome. Nephrotic syndrome is caused by whom? It is caused by Plasmodium malariae. Highlight M and N and write down. M and N are consecutive letters in English. Okay. M causes N, malaria causes nephrotic syndrome, or N caused by M, nephrotic syndrome caused by malaria. Then next we need to know about something known as reactivation malaria. Reactivation malaria. It is not reinfection, it is reactivation. It is of two types, relapse and recrudescence. Relapse and recrudescence. Now, relapse is seen in case of Plasmodium vivax and Plasmodium ovale. Highlight V and O, vivax and ovale. And recrudescence is seen in case of Plasmodium falciparum and Plasmodium malariae. Highlight F and M. Alright. Now, relapse occurs because of what is known as hypnozoites. Hypnozoites persisting in the liver. Now, who are these hypnozoites? This is my own symbol for liver. Okay. See, some of the sporozoites, when they reach the liver, they do not initiate the pre or erythrocytic cycle. Instead, they transform into dormant forms known as hypnozoites. Okay. Remember, hypnozoites are hypnotized forms in the sense that they are sleeping dormant forms of the parasite, and they can wake up from this sleep later on, and they can again precipitate an attack of malaria. Alright. So, what is meant by waking up from sleep? These hypnocytes can initiate another round of pre or exoerythrocytic cycle, and then again the entire chain of events will happen and the patient will develop symptoms. Now, what is recrudescence? It is caused by what? Recrudescent malaria is caused by, it occurs because of the drug resistant forms present in the blood. Drug resistant forms of the parasite present in the blood. Okay. Sometimes the organism develops resistance to the anti-malarial drugs, and therefore the parasite is not cleared off from the circulation. It persists, and this is seen in case of F and M, falciparum and malariae. Now guys, next we have the diagnosis of malaria. Diagnosis of malaria. Now remember the gold standard method. The gold standard method of diagnosis of malaria is what? It is peripheral blood smear. It is peripheral blood smear, that is PBS. It is of two types. Thick smear and thin smear. Thick smear for diagnosis and thin smear for speciation. Okay. Thick smear will help you diagnose the presence of Plasmodium genus, and thin smear will help you comment on the type of species. Okay. So what all things you need to see on a peripheral blood smear? Make 5 columns like this. Now guys, whatever I am highlighting right now, listen to the statement very carefully. I have written some letters in orange color: V, F, M and O. Remember from from left to right, V, listen to FM, that is fast music. O should remind you of 0, keep it in the last column. Okay, just leave it alone in the last column. Now, we listen to FM or fast music from left to right in the sequence only you are supposed to remember. Now, the first character is the first differentiating point is about the percentage of RBCs invaded. The percentage of RBCs invaded. In all cases, it is less than 2%, except falciparum, which is the most severe one, more than 2%. Okay. Now, the stage of RBC invaded. The stage of RBC invaded. Remember, falciparum, I earlier on also mentioned that it attacks all the stages of RBCs. Okay. Now, vivax invades the young forms, the young RBCs, and also the ovale species invades the young forms. Okay, young RBCs. Make an arrow like this. Okay, first and last young. Okay. Then what about malaria? It invades the old RBCs. Okay, just a little tip. Remember, M is malariae. Mal means old. Mal means old. Malaria is going to attack or irritate or disturb old RBCs. Okay. So remember this regarding the stages infected, stages of RBCs invaded. Alright. Then guys, there is something known as stippling. Stippling means what? Stippling means, the presence of pink or red colored dots on the surface of the infected RBCs. This is just a schematic diagram. For example, if this is an RBC, okay. When properly stained, some pinkish to reddish dots will be seen on the surface of the infected RBCs. Okay, so pink or red dots on the surface of the infected RBCs, they are seen when stained properly, and they represent what? These dots actually represent membrane bound structures. They represent membrane bound structures in the infected RBCs, which help in protein transport from the parasite to the erythrocyte surface. Okay, to the erythrocyte surface. So remember, these are pink to red dots seen on the surface. membrane bound structures they represent in the infected RBCs, and what is the job? Protein transport from the parasite to the RBC surface. Now you need to know the names of these structures: S, M, Z and J from left to right. Schuffner's dots, Maurer's dots, Zeeman's dots and James dots. So far in this table, three important characteristics I have told you: percentage, stage and stippling. Now continuing further in the table, remember from left to right we have what? We listen to FM. Alright, now guys, we have covered the life cycle, and what all we saw in the life cycle, what all stages of transformation we studied. The early trophozoite, that is ET oblique the ring stage, then the late trophozoite, alright, then schizont and gametocyte. Now all these, this tick mark represents all these are seen on PBS in case of Vivax. In case of malaria also, all four are seen. In ovale also, all four are seen. In falciparum on the other hand, early trophozoite or ring stage and gametocyte are seen, but not late trophozoite or schizont. This is very important to know. Why is that so? Before I tell you the reason. Do you think I have missed any stage in this table? Yes, I have deliberately missed it, merozoites. You know why? Because merozoites are too tiny to be seen on peripheral blood smear. Okay. So note down, merozoites are too tiny to be seen on peripheral blood smear. Let me tell you now the reason why these two forms, late trophozoite and schizont, are not seen in case of Plasmodium falciparum. In case of Plasmodium falciparum, what happens actually? The LT, that is late trophozoite and schizonts. Okay, these forms, they attach to the endothelium. They attach to the endothelium of the capillaries of the internal organs wherever they are getting formed. Okay, so these forms, they are going to attach to the endothelium of the capillaries of the internal organs wherever they are formed. Okay, this is especially seen in case of kidneys and brain. So these forms, because they are present in the they get stuck onto the capillaries, they do not enter the peripheral circulation. And in diagnosis of malaria, the gold standard, as I said, is peripheral blood smear. So when they are not going to appear in the peripheral blood peripheral circulation only, they are not going to be seen on peripheral blood smear. Okay, so this leads to what? This is called as cytoadherence. This is known as cytoadherence. And cytoadherence, this leads to, therefore, sequestration of the parasite in the internal organs. Alright. A common question asked is, what is the reason behind these forms not being seen on PBS? And two confusing options are, one is cytoadherence, the other is sequestration. Now what do you think is a better answer out of the two? Better answer is cytoadherence, because sequestration or hiding of the parasite is a result of cytoadherence. So the primary reason is cytoadherence, which in turn is going to lead to sequestration. So you have to mark in such questions cytoadherence as your answer. Okay, unless and until there is heavy parasitemia, which will facilitate the entry of these morphological forms into the peripheral blood, they otherwise are not seen in the peripheral blood, and hence they are not seen on PBS. Right. So next you need to know, if suppose this is an RBC, if you see multiple ring forms in the RBC, if multiple if multiple ring forms are present, it means it is Plasmodium falciparum, PF, Plasmodium falciparum. Alright. Then if the ring form is stuck onto the RBC membrane, then it is referred to as apple Q form or a coal form. Alright. If suppose you are able to appreciate this rectangular band like structure in the infected RBC, this is called as what? Band form. What do these what does this band form represent? It represents the LT or the late trophozoite of Plasmodium malariae. Okay, late trophozoite of Plasmodium malariae. So remember this. So this is about the peripheral blood smear, guys. Guys, have a look at this. Plasmodium falciparum. Okay, now we'll go from left to right, A, B, C. A is showing what? This is a thin blood smear showing the different forms of Plasmodium falciparum. Figure A shows the multiple ring forms. As you can see, let me point it out. See this arrow, this black arrow, multiple ring forms are seen, and the ecole forms, okay, ecole or apple Q forms of Plasmodium falciparum. Then in the middle, you see double dot form or headphone shaped ring form, okay, double dot. This again is a feature of Plasmodium falciparum. Remember, double dot or headphone shaped ring form, two dots seen. Then very importantly, the gametocyte shape of Plasmodium falciparum is spherical. Figure C, this one is the gametocyte. The third one, falci means what? Falci means sickle shaped. Alright, that is why the species has been named so, because the gametocyte is like this, sickle shaped, whereas the gametocytes in other three species are spherical. Okay, then this is picture of vivax, a thin blood smear showing different forms of vivax. Figure A is the ring form. In the center, figure B shows the gametocyte spherical, and figure C shows the multinucleated schizons. Alright. Then guys, this is the thin blood smear showing the ring form of Plasmodium malariae. That is the band form. Okay, some books mention it ring form, but late form, late trophozoite is a more appropriate term. Okay, so remember the gametocyte. The gametocyte of Plasmodium falciparum is like this, sickle shaped, whereas the gametocyte of other species, other three species, is spherical. So this is about the peripheral blood smear. Then one can go for quantitative Buffy coat, that is QBC, where acridine orange stain is used to detect the nucleic acid of the parasite in the infected RBCs. Remember, RBCs do not have any nucleus. Okay, so we can make use of a stain which stains the nucleic acid, that is acridine orange stain, to detect the nucleic acid of the parasite which is there in the infected RBCs. Then antigen detection. Antigen detection, HRP2, that is histidine rich protein 2, and LDH, lactate dehydrogenase. HRP2 antigen is present only in Plasmodium falciparum, whereas LDH is present in all the 4 species. See guys, because falciparum causes severe disease, it's very, very important to diagnose it properly, timely. That is why an antigen specific to Plasmodium falciparum is used in the kits used for diagnosis. So in HRP2 detection will help us diagnose malaria because of Plasmodium falciparum, but this is not used for prognosis, because HRP2 levels will continue to stay positive for weeks after recovery, whereas LDH levels fall with treatment. Therefore, LDH levels can be used to follow up the patient, therefore used for prognosis.